Hello friends, welcome back to my YouTube channel Astin Padasale. In this video, we are going to discuss top 25 important highly expected questions for this DBT beta examination. First question, if the DNA content of a diploid cell in G1 phase of cell cycle is X, then DNA content of same cell at metaphase of meiosis 1 will be. What is the DNA content? Like that they are asking, and they had given what is the DNA content in metaphase of meiosis 1. So we all know the answer. The DNA content will be double because in between the G1 phase and metaphase there is a phase called S phase, which is called the synthesis phase, and in that our DNA will be getting replicated. So DNA content of same cell and metaphase of meiosis 1 will be 2x. Okay, so this is the formula sheet, friends. Here N means number of chromosomes, C means DNA content. So in G1 phase the DNA content is, if it is 2C means, and ultimately in metaphase 1 of meiosis 1 the DNA content will be double. Okay, because it is in between, there is a S phase. So first you need to know whether they are asking number of chromosome or whether they are asking to find a DNA content. The question they had given DNA content, so the correct answer is 2x. Okay, so this is the formula sheet, friends. So here I had informed you the G1 phase the DNA content in 2C, whereas here metaphase 1 of meiosis 1 is 4C, which means the DNA is doubled. Next question, which of the following is the ideal molecular marker of mature lysosome? So mature lysosome will be having a mannose-6 phosphate receptor. Okay, so those protein or those enzyme that need to enter into lysosome will be having a mannose-6 phosphate tag. Okay, so you should know about some of the signal sequence, like if a protein need to enter into ER means then it need to hold this particular signal sequence, which include 3 successive leucine, valine, glycine, isoleucine, leucine, threonine, tryptophan, alanine. Only then the protein can able to enter into ER. These are a barcodes or some kind of tag that we can able to tell. Okay, next a protein need to present in the lumina of endoplasmic reticulum means then it need to have this particular KDEL. K for lysine and D for what? ASP. That is aspartic acid, and E for glutamic acid, L for leucine. If a protein need to enter into mitochondria means then it need to hold arginine and followed by few important amino acid, again arginine, lysine and arginine. Okay, next if a protein need to enter into nucleus means then it need to hold this proline, lysine, arginine, lysine, valine. If a protein need to enter into peroxisome means it need to hold this particular sequence, which include SKL, that is serine, lysine and leucine. Next, glycosylation of protein occurs in. So, glycosylation occurs in endoplasmic reticulum. So, there are different types of glycosylation. N-link glycosylation will be occurring in endoplasmic reticulum. Okay. So, what is mean by N-link? So, here the glycan group will be binding to the amino group of your asparagine amino acid. Next, second type of glycosylation is O-link, which means a monosaccharide will be binding to OH group, that is hydroxyl group of this serine and threonine. And this O-link glycosylation not only occurs in ER, it can also occur in Golgi plasm, Golgi reticulum, cytosol and nucleus. Next is glycation. What is mean by glycation? Here the glycan core will be linking your phospholipid and protein. So this is an important process which will be helping to hold glycolipids like GPA anchor in the plasma membrane. Okay, next is sealing glycosylation, which involve here in this particular sealing glycosylation your mannose will be binding the indole group of your tryptophan amino acid. Next is phosphoglycosylation, where your glycan will be bind to the serine amino acid by forming a phosphodiester bond. Next is higher version of blossom can be used to detect. So as we discussed already, blossom matrix with high number and PAM matrix with low number will be generally employed for comparing closely related sequence, or in otherwise we can able to say it is employed for determining or comparing less divergent sequence. At the same time, we can also employ blossom matrix with high number and PAM with low number for comparing more conserved sequences. So here the question is the higher version of blossom can be used to detect closely related sequences. So kindly take down the notes of this particular and just try to remember each and every line so that it will be very much helpful. Next is if a man of blood group AB marries a blood group woman of blood group A. So the father blood group is AB and mother blood group is A. Now they are asking what about the blood group of children. Okay, so we can able to simply put a Punnett square. So father blood group is AB, therefore his genotype will be IA, IB. Mother blood group is A. There are two possible genotypes. Either she might be a recessive, that is IA and small I, or she might be a homozygous dominant, which means her genotype may be IA and IA. If you do a compare Punnett square, there will be a possibility of three blood group. Either the child will be A blood group or AB blood group or B blood group. If you don't know how to put this Punnett square means, just remember this particular formula sheet. If both the parent are having a same blood group, like say if a father is A and mother is A means, then ultimately only two possible blood group are possible, which include A and O. Next, if both the parent blood group are B means, then again the children will be getting either B blood group or O blood group. If both the parent are O blood group means, then ultimately there is only one possible blood type for children, that is O blood group. If both are AB means, then the children will be getting A blood group or B blood group or AB blood group. Okay. While in this case, that is, if father blood group is A and if mother blood group is B means, then all the four blood types are possible. In a question they have given father blood group is AB and mother blood group is A. Therefore, the possible blood group include A, B and AB. Say if one parent blood group is AB and another parent blood group is B means, then again three blood groups are possible, which include A, B and AB. This is an easy thing for you to remember. Next question, which of the following enzyme is not used as pyrosequencing? So pyrosequencing is just a kind of sequencing. It is an advanced mode of DNA sequencing. It helps to know the sequence of a particular DNA. Okay, so which of the enzyme is not used? So you should know there are four enzymes that are required for pyrosequencing. First enzyme is fragment of DNA pol 1. So they are using DNA polymerase 1, not a DNA polymerase 3, which is generally employed for your usual DNA replication. The second enzyme is ATP sulfurylase, and third enzyme is luciferase, and fourth enzyme is apparase. So fragment of DNA polymerase is involved in addition of your DNTPs, whereas in DNTP is added, it will be releasing a pyrophosphate. This PPI means it is called as inorganic pyrophosphate, and this PPI will be serving as a substrate for second enzyme called ATP sulfurylase, which will be generating a ATPase, and third enzyme is luciferase, which will be converting the luciferin to oxyluciferin, ultimately generating a light. Okay, this light will be detected so that the DNA sequence will be, you can able to find out the entire sequence of your DNA. So what about this apparase enzyme? This apparase is involved in removing a unincorporated nucleotide and unincorporated ATPs. Okay, it's kind of garbage disposal system. So here DNA polymerase is involved, fragment of DNA polymerase is involved, luciferase involved, ATP sulfurylase involved. With regarding to this, pyrophosphatase is not involved. If this particular enzyme is added means, then ultimately the pyrophosphate will be, we can't able to determine the pyrophosphate, simply cleave the pyrophosphate. So there are three enzymes used in pyrosequencing, which include fragment of DNA pol 1, ATP sulfurylase, luciferase and apparase. Next is administration of DTP vaccine, which stimulates which of the following type of immunity? See, adaptive immunity is classified into natural and artificial, and natural is again classified into passive, active. Okay, so for example of adaptive, natural, passive, adaptive immunity include your maternal antibodies that we are getting through the placenta, or a breast milk that a child will be getting immediately after a birth. So next example of active natural adaptive immunity include your infection. We had exposed few important natural immune infections. So those kind of immunity is an example of a natural active immunity. Next example of artificial passive adaptive immunity include your antibody transfer, like monoclonal antibodies and some other antibodies that are transferred from one person to another person or from few other animals to other animals. Okay, the example of artificial active adaptive immunity include your immunization, which include vaccine. So the correct answer for this particular question is that vaccine is an example of, it will be stimulating artificial active adaptive immunity. Okay, so next, which one of the following codon is used for selenocysteine during protein biosynthesis? So selenocysteine is a 21st amino acid and pyrrolysine is a 22nd amino acid. Okay, so the code for the selenocysteine is UGA and the code for pyrrolysine is UAG. Okay, so here which particular codon will be coding for the selenocysteine include, it is UGA, whereas this UAG is coding for your pyrrolysine. Next is SSR is said to be a DAS type of molecular marker. So molecular marker are classified into two types. One is called as dominant marker, another one is called as codominant marker. So dominant marker is just, it will be telling whether the allele or whether the gene is present or not. It will not show any allelic pattern, whether the allele is a homozygous dominant or homozygous recessive or heterozygous. It will not give any such detail. An example of dominant marker include REPD, AFLP, EST, ISSR. Whereas codominant marker will be clearly showing whether the allele is following a homozygous dominant pattern or whether the allele is a heterozygous recessive or whether the allele is a heterozygous state. Like that, they will be telling you a detailed explanation will be given. The example of codominant marker include RFLP. So you should know AFLP is an example of dominant marker and RFLP is an example of codominant marker. And this SSR, SEAR, SNP, STs are coming under this codominant marker, which will be clearly showing a band homozygous dominant, homozygous recessive and heterozygous. So SSR is an example of codominant marker. Next is which of the following cell surface marker is used to identify the B cell from the blood sample. So you should know this is an important question, friends. Okay, so let me tell you the CD marker for B cell include CD19, CD20, CD21, CD40, which are generally employed for analyzing your B cell. These are B cell marker. Okay, this will be present on the surface of your B cell, whereas CD16, CD56, CD94, CD158, CD161 are often selected for analyzing this NK cell function, which means these CD, that is cluster of differentiation marker, are present on the surface of your NK cell. These are the CD antigen for your identifying a T cell. If you take CD2, it is present in all T cell and all NK cell, whereas CD3 is present only in mature T cell, and CD4 is present in your T helper cell, and CD8 is present in your TC cell, and CD25 is present only in activated T cell. Same, the CTLA4 and CD40L is also present only in activated T cell. Whereas if you take the CD28, it is present in almost all CD4 T helper cell, and it is present in 50% of population of the CD8 T cell, and in addition it is present in plasma cell, and it is also present in activated B cell. So here the question is that which of the cellular marker is used for identifying B cell from blood sample, and ultimately the marker which is used is CD19. Rest of the marker like CD3, CD4, CD25 are generally employed for identifying T cell from your blood sample. Next is Brifildin A inhibit protein transport. So this particular antibiotic inhibit the transport of protein from ER to Golgi. So you should know if a protein is transported from endoplasmic reticulum to Golgi, that particular transport is called as anterograde transport or forward transport, whereas if a protein is transported from Golgi to ER. So few protein after completing the post-translation modification in Golgi, if a protein need to enter into ER means then it will be following a retrograde transport. So those protein that are resident of ER, endoplasmic reticulum, or those protein that need to function in nucleus will be taking this particular retrograde transport. Okay, so this particular Brifildin, it will be inhibiting the anterograde transport, or it will be inhibiting the transport of protein from ER to Golgi. So kindly know the important inhibitor which are disturbing the protein to enter into ER. At the same time, few other this particular thing are called as protein maturation inhibitor, which means they will be disturbing a process of post-translation modification that are occurring in endoplasmic reticulum. Okay, next question is tunicamycin block. So this particular tunicamycin block N-link glycosylation. So here also N is there and here also N is there. So it will be specifically blocking N-link glycosylation. What about O-link glycosylation? So benzyl N-acetyl alpha galactosamine is involved in blocking. They are inhibitor of this O-link glycosylation. Next is GTP binding protein are inactive in GTP bound form. They are active in GTP bound form. Which of the following protein converts the GTP bound form to GDP bound form? So first you should know GTP bound form of protein is called as inactive form, and the GTP bound form of protein is called as active form. Okay, so with regarding to the GEF, which is called as guanine nucleotide exchange factor, is involved in converting the inactive form of this particular protein to active form, which means they are called as exchange factor. So for the GTP, they will be exchanging GDP. If you want to convert the GDP bounded inactive protein to GTP bounded active protein means then in the presence of GEF is required. Whereas if you want to switch off the mechanism, if you want to convert a GTP bounded active form into GDP bounded inactive form means then in that case you need GAP. GAP, that is gap. What is meant by GAP? It is GTPase activating protein, which will be cleaving this GTP by a release of phosphate molecule. It will be converting the GTP to GDP. This is involved in your GTP hydrolysis. So here they are asking which particular protein is involved in converting your GTP to GDP. Then ultimately it is GTPase activating protein. Next, if a nucleotide sequence encoding a protein is known, so you are having a query sequence which is nucleotide and you want to find a homologous protein. So you want to find a nucleotide query and you want to search this nucleotide query in a protein protein database, which particular BLAST program you will be employed. So first BLAST-10, which is employed for searching a nucleotide query against a nucleotide database. BLAST-P, which is for searching a protein query in a protein data bank or protein database. Next is with regarding to this T-BLAST-10, which means if you are having an amino acid sequence and if you want to find the matching nucleotide sequence, means in a particular data bank, means you need to employ this T-BLAST-10. So T-BLAST-10, when you need to employ, if your query type is protein and you want to search your amino acid sequence or respective nucleotide coding sequence, means then at that time you need to employ this particular T-BLAST-10 program. Whereas what about this BLAST-X? You are having a nucleotide query sequence and you want to find a homologous protein sequence in a database, means then you need to employ this BLAST-X. So here they are having the query sequence is nucleotide and the data bank sequence is protein, and at that time you need to employ BLAST-X program. Okay, when you will be employing BLAST-N, when you are having a protein query sequence and you want to search in a nucleotide data bank, means then at that time point you need to employ T-BLAST-N. Whereas if you want to compare a nucleotide to nucleotide, that is 6-framed translated nucleotide sequence to a 6-frame nucleotide translated sequence. Like if you want to compare a protein and protein for which only you have a nucleotide sequence, means then at that time point you need to employ T-BLAST-X program. Okay, so the rest of the question I will be discussing in my second part of the video. So thank you friends, thanks for watching this video.